Hello everyone. This is our new DNA extraction kit. Our last DNA extraction kit has finished. So today I will open this. Let's unbox this Zymo kit. So let me first introduce you what's written over here. Quick DNA mini prep plus kit. And this is the information. Made in USA, zymoresearch.com, and these are the sides of this kit. See what's written: imagination is more important than knowledge. Knowledge is limited. Imagination encircles the world. Albert Einstein. Right? Now I will open it and we will explore what are the reagents that are provided in this kit. Let me open it. Right? This is the scissor and I'm opening it in this way. Yes, so this is the kit, right? So we have been provided these tubes. These are the collection tubes, and these are the different reagents. gDNA wash with this 400 microliter as instructed in the manual. These are also the filtrates, right? These are also used in the DNA extraction protocol. So let's start with some of the enzymes. So this is proteinase K. This enzyme is a key player in the initial lysis step. Its purpose is to digest proteins, including nucleases. The kit optimizes protein K, ensures rapid and effective protein degradation. Then this one, genomic binding buffer. And what is the function of this buffer? That this buffer, it often supplied in different version, biofluids and cell buffers or solid buffers. So you can use this one, right? These are the reagents. And then we have solid tissue buffer, this one. Then we have this DNA elution buffer. The final step, it involves eluting the purified DNA from a spin column. So these are the spin columns, these are the collection tubes. These are used during the DNA extraction. Then we have this DNA pre-wash. This wash buffer is used to remove proteins, salts and other large cellular debris that did not bind to the column matrix. Okay, then we have DNA pre-wash buffer. So this is given in larger quantities. Then yes, of course, I think this time there is no manual present. So I'm not promoting this DNA kit, but it's like a review. I usually prefer this one for the extraction of DNA. It can help you extract DNA from different samples such as mammalian cells, solid tissues, biological fluids. Even you can extract DNA from a sperm cell as well. We can use extraction of from the blood cells using Kaizen kits as well, but I usually this time I'm using this one for the extraction of almost 400 samples. So you can extract around 200 samples from this kit. So this is the quick DNA mini prep. So these are the reagents, collection tubes and other items. So you can see there are different proteinase K is present there. They have given you in huge quantity. So this is the powder form you will make it. This is 20 milligram. Right, you can see this one, 20 milligram. This is the cat number. So let's start our DNA sample from today. Thank you for watching this.